Okay, so this is at the end of the slides that I posted today on Blackboard. So the slides today are called like two-way design and Sexy Stroop 1. That's what we're going to be talking about today. And this is at the end of those slides, but it's important, so I'm just going to talk about real quick here at the beginning. There's several things we need to remember to take care of in the class. Groups, we need to sign up for groups. I feel like most of the people that are attending the class synchronously have signed up for groups, but it's the people that are not attending synchronously that are lagging a little bit. As of an hour ago, there was only 24 people in the class that had signed up for groups. So there's definitely a handful of people that still need to sign up. I would glad to be help match people into groups. Just contact me and we can do that. The Google Sheet is right there. Just click on it. Please also remember that your Zoom recording for Lab 2 is due tomorrow night. The instructions for that were provided previously, and there's videos I posted the links on Blackboard explaining that. If you need help, I'm happy to meet to help you with any aspect of that. I met with a student earlier today who's having some coding issues, and I believe we were able to work them out quite well using just Zoom. That's due tomorrow night. Don't forget about it. Let's see. Also, do Sunday is your first official Group Project 1 assignment. It's an easy one, and really it's meant just to get you guys together and verifying that you guys have met at least one time as a group. I have given the instructions previously, and the instructions are also on Blackboard for this assignment. Basically, you just have to record a meeting amongst yourselves and then share the entire folder with me as a link. And then you're going to turn in that folder, that link on a Microsoft Word document into Blackboard. Please note that for Lab 2, I had asked you to just send me the email with the link. Which works, but I've decided after doing that it works much better if you guys send the link as a Microsoft Word document. So this one, you can just send me a link for Lab 2 recording. But for the entire folder, which I want for Group Project 1 Zoom recording, go ahead and put a link on the Microsoft Word document to the entire folder. Make sure I am able to access the contents of that folder, and then you'll submit it on Blackboard instead of emailing me. Don't email me those links anymore. I will always want you in the future to turn them in as a Microsoft Word document where you copied and pasted the link into the document. Question? Yeah. Is that a
Um,
question? Yeah.
yes, it did. Sorry. For the recording for the Group Project 1, so in the folder, you just need the Zoom of us just saying our names, and that's all you need in the folder, correct?
Well, the folder I want everything that Zoom makes by default in the folder. So that could work. All I might need to see is the video. However, if you have a group member who does not have a microphone on their computer, so they have to talk using chat, and they would probably also not have a video camera as well. If there's a person that has to communicate with you using chat, that's okay, but I'm not going to see their chats in the recorded video. So if there are chats that occur during the recording of a video, Zoom will by default include a text file in the folder, and it will include all of the chats that occur during the recording of the video. It kind of records the chats on a text file. And so the video might work just by itself, but if there are people in your group that cannot say their name, they can't say it into a microphone and me hear it, then they're going to have to chat their name. They'll have to say, I am so-and-so, and then I'll have to see it on the text file. So my suggestion is everyone should just share with me the content, the entire folder that Zoom makes, which if there are chats, it'll include the text file. If there are not chats, it will not include the text file. Does that make sense?
Yes.
Cool. All right. Any other questions about that? Okay. The researcher assignment, I want an update. I checked. It was formally due Sunday, but I'll accept them late. Especially because, well, as of now, there's only 13 researcher accounts that have been created. But I talked to, Phil emailed me this morning, and he mentioned that he's a little bit behind on verifying researcher accounts. And he told me he has received a large influx of requests. and he's gotten a little bit behind on them. I am sorry about that. He said he will try and get on it extra quick because I kind of nudged him nicely to try and speed up a little bit on that. So hopefully he'll be able to get those done this weekend. But please make sure, I have given points on Blackboard for the 13 students who I do have researcher accounts. I have given you the points on Blackboard. You get a 10 out of 10 if you have a researcher account. If you do not have points, make sure that you've done everything you need to do to get the researcher account. Basically what that is you have to complete the SONA subject pool use quiz. If you've completed the SONA subject pool use quiz, that's really all you have to do. There's some steps you have to do before completing the SONA subject pool quiz. But once you complete the SONA subject pool use quiz, then you should be done with everything that you need to do for the researcher account. So make sure that you've done that. And I should mention there were some students that I've talked to that have a SONA participant account, which is what you did if you took 104, the intro class, when you were participating in studies. That's different than a researcher account. A researcher account gives you access to things that you do not have in a participant account. So please don't get confused into thinking you already have a researcher account just because you have a subject account. They are definitely different. And finally, I will be sending out some formal information about Group Project 1 this weekend. I think I have some things in the notes here. Yeah, and just some things that I'm going to tell you. I mean, I already know what we're going to be doing for Group Project 1. I will send you information regarding the details this weekend. I will tell you once again, half will be running participants. Half of them will listen to Mozart music, and the other half will do something else. They'll either sit in silence or maybe they'll listen. Someone suggested white noise last class. In the chat, I looked at it at the end. Someone suggested the control group could listen to white noise, which might work, though. I think the white noise might drive them crazy listening to static for 10 minutes. But we'll have to do something for the control group where they're doing something other than listening to music. More suggestions are welcome. So that's what we're going to be doing, and then they'll listen to music or they'll sit in silence or some control task for 10 minutes. And then everyone will take in this experiment, will take the Stroop task, and we're basically looking at, does listening to Mozart music, does it influence people's performance on the Stroop task? You are going to have to code that experiment in PsyToolkit. And the thing that I send out this weekend, I will give you some information about how to code it. But at this point, you know the basics of PsyToolkit. If you did Lab 2, then you should know the basics of PsyToolkit, and you should have also watched those videos, which I linked to previously, on how to code experiments in PsyToolkit, which basically teaches you everything you need to know in order to do the experiment that we're going to do for Group Project 1. Of course, if you run into trouble, more than happy to help with that. But so I'll give you some information about what you'll be coding this weekend and the information I send you, but you'll also have to be independent and figure some of this stuff out using the videos that I've already given you access to. I've given you the link and showed you where those videos are available on the web. Let's see. So you'll have about 3 total weeks to code, that is to write the code for your experiment. You make sure the experiment works, and then to collect data from your participants. All groups will be collecting data somewhere between 6 and 8 participants. If you're a group of 3, you'll collect data from 6 participants. If you're a group of 4, you'll collect data from 8 participants, basically 2 participants. You'll collect data from 2 participants for each participant in your group, for each group member. And you'll basically have 3 weeks from this weekend to collect all the data. So you have to make the experiment inside Toolkit and collect data, and you'll have about 3 weeks to do all. At least that's the plan right now, is that maybe I'll show you the syllabus here. Where is it? Data collection is due toward the end of week 6. My current plan is to make it due that weekend. So probably like Sunday night of that is when data collection is going to be due. Right now, this is us. Over this weekend is when I'm going to send you information. So you have week 4, week 5, and all of week 6 to create the experiment in SciToolkit, and then to collect the data from participants. And in the instructions that I send you this weekend, I will give you some information about how to actually run the participants in SciTool, how to actually collect the data from participants using SciToolkit. I will mention, we've already started coding the experiment. Assuming that you have completed Lab 2, which now a lot of people have completed Lab 2, that is basically the skeleton for what we're going to be doing for Group Project 1. You'll only be making you'll have to attach the experiment that you've already made to a survey, which is not really that difficult at all. And you'll have to make a couple slight modifications in the experiment itself. But you've already done the hardest part of, I would say, the hardest part of the programming the experiment because of Lab 2. You'll just have to make a couple more slight modifications and you'll be ready to roll as far as collecting data from participants, which you'll be doing in about 3 weeks. I will point out that you will have Group Project 1, the first half, which is basically you're going to be writing the introduction and some of the methods. That's basically what Group Project 1, that will be due in a little over 2 weeks from when you receive the information this weekend. So this next 2 or 3 weeks is pretty crucial regarding Group Project 1. Okay, so that's it all about things to take care of. Any questions or comments about that stuff? All right. So today we have you have a lot of kind of assignments that are that we have to take care of still. So really my goal today, I do have some lecturing I'm going to do on these 2 things. The two-way design, which should actually be review for most of you. I've talked with the methods teachers, and my understanding is that basically all of the methods courses previously have talked about this topic before. So this topic of two-way factorial designs, that is main effects and interactions, this should be review for most of you, assuming you've taken research methods, which I believe you all have. So this should not be particularly hard. I'll talk about that for a while, and then we'll talk a little bit about psyc625 1, which is the article that you have due for today, and then that's it. And I don't think that's going to take the entire period. Hopefully you'll have some time, class period, to work on some of these assignments, and of course I'm always happy to help meet with students over Zoom if you do have questions about anything. Starting talking about two-way factorial designs. And I actually want to begin by talking about an example of something which is just a one-way design. This is the first article you read for this course. This is from Mozart 1. And in this study, there was only a single factor. They called it listening condition, a one-factor repeated measures analysis of variance. So this a one factor indicates that there was only one factor or one independent variable in this study. So it was a one-way design. Another example, which I've written in the notes portion, the Stroop task that we did, lab 1, the very first lab, was another example of a study which is a one-way design. That one was a one-way design with only one factor with 2 levels. This one right here is a one-way design, a single factor with 3 levels, 1, 2, 3. The Stroop task from week 1, we had one factor congruence, and then one of the levels was incongruent, the other level was congruent. So that's 2 examples of studies, the Mozart study here, and then the Stroop task, which we did lab 1, where there's only a single factor. And in this lecture today, we'll be contrasting that with factorial designs, which I have already mentioned before. A factorial design means that there are 2 or more factors. So for this next part, here it says, well, why would we use complex designs? And the truth of the matter is that in psychology, the majority, the super majority of studies use complex designs. The studies like Mozart 1, which was published in the great journal Nature, where there's only one factor, those are actually pretty rare these days. They're more common back in the past, but these days, the study designs usually involve at least 2 factors, maybe even 3 or more factors. But that's because behavior is complex, and there are usually going to be a lot of factors that are involved that are influencing a given behavior. just in general, studies these days use 2 or more factors, as almost most studies are just that way. Here's some examples of studies which are using complex or factorial designs. So it says, let's see, maybe hot temperature increases aggression, but only following a frustration. So we need to think, in this little scenario here, what would the factors be and what would the dependent variable be? And so I'll kind of help us out with this one. One of the factors would be the temperature. So you would have a group of participants. Some of them would be like in a room that had a hot temperature, and another people would be in a room that was low temperature or room temperature. So that's one factor. The other factor would be whether people had just experienced something that was frustrating or not. And there are laboratory experiments, where the researcher will purposefully frustrate half the participants, and then the other half of the participants, the research does not frustrate them. So one factor would be temperature with 2 levels. The other one would be amount of frustration, which would also probably have 2 levels. And then can anyone tell me what would the dependent variable here be in this study? Can someone tell me in this little scenario, what would the dependent variable be?
Aggression.
Aggression is correct. By the way, you might have seen these examples might be familiar. I stole about half of the examples that I'm using today. I've stole from another professor in the department who teaches research methods, including these examples right here. Some of you might have seen these. So if these look familiar, that is the reason why you saw them. Some of you saw them previously in research methods, likely. So another example, maybe ethnicity predicts an individual's self-esteem differently depending on their socioeconomic status. So one factor, which would be a quasi-independent variable, because you cannot randomly assign people to be in different ethnicities, would be their ethnicity. So you'd have different people of varying ethnicities. And then those people would also, another quasi-independent variable would be their socioeconomic status. So some people would be like high socioeconomic status, and some people would be low. Maybe there would be middle socioeconomic status. Those are the 2 factors. And the dependent variable here would be self-esteem. So how do these factors, ethnicity and socioeconomic status, how do they interact, or do they interact to influence a person's self-esteem? These are examples of complex designs or factorial design. Once again, factorial design means 2 or more factors, which is basically what complex design means as well. Questions or comments here? So we're kind of going to go into this experiment here. This is an actual published experiment. In case you're curious, I have the abstract right here. I copied and pasted it from the internet. So it's a social psychology experiment. It's an example of a 2-factor study. And it illustrates some of the basic concepts about 2-factor studies that are important to know. So the study here has to do with eyewitness testimony and some really interesting research that basically shows that certain factors can influence whether a person misremembers something about a crime that they witnessed. So in this study, everyone at the beginning of the study watches a video of I believe it was a bank robbery, of a crime being committed. And then later on, there's 2 factors that participants are exposed to. So the factors are the thing that they're interested in this study is whether receiving misleading questions, whether that influences your memory of the crime and your perception of the expertise of the individual who is asking you those questions. So once again, we have 2 factors here. One factor, I guess they're down here is where they are. The type of question is one factor. So whether it's an unbiased or misleading question. So I have those in the notes here. You can see them. So examples of misleading questions are written normally. The ones that are unbiased are in parentheses. So what kind of gloves were they wearing? That is a misleading question compared to were they wearing gloves? Because if you ask a person what kind of gloves were they wearing, that implies that indeed the people who were in the video or committed the crime were wearing gloves. Similarly, what did the other guy's gun look like, compared to an unbiased question, which be, did the other guy have a gun? When you ask a person, what did the other guy's gun look like? That implies that indeed the individual had a gun, when in reality, he or she, the other guy might not have had a gun. So those are, and there's a couple other examples. I won't go through all of them, but that's type of question, whether the question was misleading or whether it was unbiased. And then the other factor, the other factor is questioner type. Whether where in the experiment, basically, there would be you watch the video of the bank robbery, and then a person asks you questions about the bank robbery that you just watched the video to. And once again, those questions could either be misleading or they could be unbiased. And the person that's asked you those questions, you might have been told by the researcher that the person who is asking you those questions is very knowledgeable about the crime. That's the knowledgeable condition. Or you'll be told that the person that's asking you those questions is naive about the crime, that they have no idea, they don't know anything about what happened. And then the dependent variable is basically the percentage of memory errors. How accurate or how not accurate was your memory of the bank robbery that you watched? So I set the stage for the design of the study. And now we're going to be thinking about the means that would be compared in the statistical analysis in this study. So this is a 2x2 factorial design, and I'll go into more detail in a future slide exactly what this means. I guess I'll tell you quickly. Each number, this represents one factor that has two levels. And this number right here represents another factor that also has two levels. And you can use these numbers, this means 2x2 design. You can use this nomenclature right here to determine how many conditions, that is how many means there are in the statistical analysis that you'll be performing. And it's quite, the way of doing it is quite simple. You just multiply 2 times 2 equals 4 conditions. And you can see the 4 conditions right here. One of the factors, we'll call it questioner type, is they call it, in this example, considered factor A. The 2 levels are shown right here, whether the questioner is considered a knowledgeable person about the crime or naive, not knowledgeable about the crime, compared to the other type of the other factor type of question, whether they were unbiased or misleading questions. And then you can see this kind of matrix here that has 4 cells. And you would have a mean for the people that were in this condition, a mean amount of memory errors in people in this condition, a mean amount of memory errors in people in this condition, and the mean amount of memory errors in this condition. So we have 4 conditions. So those 4 conditions will be really important for the statistical analysis that will be performed. So in a factorial design with 2 factors, there's basically 3 p-values that will be produced by the statistical analysis, the ANOVA. Each one of those p-values could either be significant or it will be not significant. And so there'll be a main effective factor A, that is the main effective questioner type. That is, in this one, the question basically is, do we create more, are there more memory errors regarding the bank robbery when you perceive the questioner to be very knowledgeable about the crime, or when you perceive the questioner to be naive about the crime? Does how you perceive the questioner, does that influence your amount of errors that you make, memory errors regarding the video of the bank robbery that you watched? That's factor A, questioner type. There's also, and that could be either significant or not significant. And there's also the main effect of factor B, type of question. The question here is, are we more or less likely to make memory errors depending on whether the question was unbiased or whether it was a misleading question? So for each one of these main effects, we're only comparing two means. Remember, we have 4 total means, we have 4 total conditions, but for each main effect, we're only comparing, in this particular study, 2. For the questioner type, we're comparing the means for the two questioner conditions, that is knowledgeable versus naive. For the type of question, we're comparing two means for the two types of questions, which was unbiased versus misleading. And then finally, the interaction effect, it looks at all the means of all four conditions at the same time. And the interaction effects are arguably the most, usually for factorial designs, they're kind of the most, I guess I'll say interesting and arguably the most important statistical result that you'll get. Each one of these will have its own p-value. So the main effect could be significant or not, this main effect could be significant or not, and the interaction could be significant or not as well. These are all somewhat independent, the p-values, that is whether we accept or reject the null hypothesis. And here's some definitions. Once again, you've probably seen these definitions before. I know personally when I took research methods in undergraduate, I was not 100% comfortable with really understanding what these terms meant. So it's somewhat helpful to review them again. So yeah, main effect, interaction defined here. Interaction, if one factor modifies the effect of another factor. To me, this is a very, even though that's true, it's a definition which is kind of hard to visualize in your head. What does it mean when you say one factor modifies the effect of another factor? I'll show you examples, which I believe will help you understand this. Another way of writing that, these are kind of similar statements. They basically mean the same thing. There's kind of two different ways of wording it. When the effect of one factor depends on the level of a different factor. So this is one interaction. These are two different ways of kind of verbally understanding in words what an interaction is, but really the best way is just to have an example and understand the example. So here are the data from this experiment that I've been talking about. Here are the means from the four different conditions right here. And we will kind of decompose the means that are compared for each of those main effects, main effect of factor A, main effect of factor B, and then the A by B interaction. So there's a new element here. So looking here, that's a simple, a basic matrix. But then I've added in the margins here, the headings here are marginal means for the main effect of A, and then marginal means for the main effect of B. I mentioned that each main effect is comparing there's four conditions here. You can see the means for each of those four conditions. But when we're dealing with main effect, at least in this study, each main effect is only comparing two means. So what exactly are those means that are being compared? So the kind of a shortcut method of finding out what are the two means that are compared for the main effect of A is just to take this number, which is the mean of this condition, and this number, which is the mean of this condition. So 13 plus 41, what does that equal? 54. And basically dividing by 2, and then you put that number right there. So I guess that would be, 27. Similarly, 13 plus 18, that equals 31. 31 divided by 2 equals this number right here. So kind of notice these are the means that are compared for the main effect of factor A, questioner type. And notice that what we're doing is in these two means, we don't care whether the type of question was unbiased or misleading. And that's why we take the average of these two numbers right here. We don't care about the difference between these two. All we care about is we want to know, was for the factor questioner type, was the person asking that question, was he perceived by the participant as being knowledgeable or as being naive? And so we collapse across these two levels right here, unbiased or misleading. We take the average of those two because we don't care about the difference between those two. So we just smash them together. Same here for the naive. We just want to know what was the average score of people who perceive the questioner person to be naive. We don't care if the questions that were asked were unbiased or misleading for the main effect of questioner type. So we just smash these two numbers together. We take the average and then we put it right here. The two means that were compared for the main effect of A are shown right there. Those are the two numbers that were compared. And then here are the marginal means, the means that were compared for the main effect of factor B. You do it the same way, but here we do it in the vertical direction. 13 plus 13, 26 divided by 2 equals 13. Let's see, what would the math be here? 59, 41 plus 18 equals 59. 59 divided by 2 equals 29.5. These are the two means that are compared for the main effect of B. That is the type of question asked. We care, was there a difference in the amount of errors that people made in their memory regarding the bank robbery, depending on whether the questions that were asked were unbiased or misleading? And for the main effect of B, we don't care about the questioner type at all. And so we smash those two numbers. This one is the knowledgeable person, and this is the naive person. We don't care about the difference between those two, so we just take the average of the two, and that gives us the 13. Similarly here, we don't care about the person who, for the main effect of factor B, we don't care about the person who is asking the question. We just care about the type of question itself. And so we just take the average of those two numbers together, which gives us this. I should mention mathematically, this strategy of just taking the average of these two numbers only works if you assume that the sample size of people in this box is equal to the sample size of people in this box. If there are unequal sample sizes, then this method does not work. But we're going to assume for this example that the sample size of all the conditions is equal, which allows us to calculate the marginal means very quickly and easily. Questions or comments so far about calculating marginal means? Okay, so is there a main effect of factor A? And apparently, yes, there is a main effect of factor A, that is questioner type, whether the person was knowledgeable or whether they were naive, because the two numbers were 27 and 15.5. Once again, I'll show you them. 27, 15.5 right here. Those numbers are different. I should mention, though, that just because these numbers are different does not necessarily mean that they're statistically significant. There has to be a big enough difference between them before the statistical analysis will say that they were statistically significant. But in this study, presumably, there was a significant difference between the means of these two conditions right here. Main effect of B, there also appears to be a significant difference because 29.5 and 13% are fairly different. The accuracy, the amount of errors about the bank robbery was definitely more after you were asked misleading questions than you were asked unbiased questions. So the more difficult conceptually thing to understand about factorial designs and complex designs is there an interaction? And so for each of the two main effects, we are only looking at the means of two conditions. But for the interaction, we're kind of looking at the means of all four conditions simultaneously. So once again, these are the terms I showed you earlier about an interaction, like how you know when you have an interaction. Kind of these two different statements are kind of equivalent. If one factor modifies the effect of another factor, or another way of saying that, the effect of one factor depends on the level or levels of a different factor. So here are the data shown in a graph of all four. So graphs, oftentimes you can see the interaction graphs kind of more easily maybe than you could in a table like I showed you earlier, a table where you write the numbers versus a graph where the numbers are represented by the height of the bars. If you look in most journal articles, you notice that most of the time the authors will present graphs like this instead of tables with numbers in them. And you can see the interaction here. You can see the interaction because there's several kind of ways to do this. I will present 2 or 3 different ways of kind of visualizing the interaction, or things that you need to kind of ask yourself to determine, does it appear that there's an interaction, statistical interaction in this graph? The way I do it is I consider this like a cluster, or I'll call it a cluster right here. These green and red bars that are in the unbiased condition right here. The green bars are when the participant perceives the questioner to be knowledgeable. The red bar is when the participant perceives the questioner to be naive. You look at this cluster and you compare it to this cluster. And the question I ask myself is the pattern of each cluster. Are they similar or are they different from each other? And so you kind of have to look at the pattern here, and I'd call this like a flat line pattern. because the heights of these bars are the same. And if you were to put a line there, it would be a flat line. But if you were to put a line here connecting these two bars, it would be like a very steeply decreasing line. And so the question is, are the patterns of these two clusters different? And you'd say, yeah, one is a flat line. Another is a decreasing line. It's like a line sloping down. And so, yeah, there appears to be a significant interaction here. Another way of kind of visualizing this or talking about it is you could say the questioner type, that is the difference between the green and the red boxes here, green and the red bars, has basically no effect or a very, very small effect when the questions that were asked were unbiased. So if I am asked an unbiased question, then it doesn't really matter as far as how that question will influence my memory of the bank robbery. It doesn't really matter whether I perceive the questioner to be knowledgeable or naive. The questioner type just doesn't matter when I'm, as far as the way my memory is influenced when the questions that are asked are unbiased. But you can see over here, another way of kind of wording this is that there's a large effect of questioner type when the question is misleading. When the questions are misleading, if you perceive the questioner to be very knowledgeable, an expert on the crime, someone that knows a lot about the crime, then you kind of, I guess you could say, put your trust in that individual maybe, or when they ask those misleading questions, you allow those misleading questions to influence your perception or your memory of the crime, the bank robbery that had happened earlier. So this is an interaction because there's a flat line here. Another way of saying flat line is basically questioner type. There is no effect here. When unbiased questions, there is no influence of questioner type. But when I'm asked misleading questions, there's kind of a negative or a decreasing line, a decreasing pattern. Another way of saying that is that there is a large effect of questioner type when the question that is asked is misleading. So this example, once again, it illustrates these ideas right here. One factor is modifying the effect of the other factor. The effect of one factor does depend on this example on the levels of the other factor. That is what an interaction is, and this is an example right here. Questions or comments so far? So here, I guess, is the third way, which is just as valid as the others, except it's probably not quite as helpful because line graphs are not as common in actual practice compared to bar graphs, which is what I just showed you. But you can graph the data. These are the exact same data, but they're shown on line graphs. So once in a line graph, the height of the lines of each point of the line represents the value of the dependent variable. Once again, the dependent variable is the amount of, memory mistakes regarding the bank robbery. And you can visualize whether there's an interaction based on whether the lines are parallel or not. And here the lines are not parallel. And so because they intersect, you would say there's an interaction. And so they both have int in them, interaction, intersect. And so that's kind of a mnemonic device. You can help yourself to know whether there's an interaction or not. Do the lines intersect? The lines that you would draw, do they intersect? Of course, no lines are actually going to be perfectly parallel. You know, when you're actually collecting data, it will never be the case that the lines will be perfectly parallel. So you always have to look at the actual p-value to determine do the lines intersect enough that we are willing to conclude that there is a significant interaction. If they intersect just a little bit, you know, they're not perfectly parallel. You have to look at the p-value to determine, are they intersecting enough that we're willing to conclude that there's an interaction. Okay, so I want to talk a little bit about this factorial nomenclature. I used it earlier, but I want to make sure that we're clear on factorial nomenclature now. So I'm going to give a couple more examples of study designs. So here is a 3x2 factorial design. So each number, once again, it says here, amount of numbers represents the amount of factors. So we have one number, we have a second number. So that means that this is a study that has two factors. The first number is the number 3. So amount of coffee, 0 cups, 1 cup, or 2 cups. That's this factor 1 right here, 3 levels. Factor 2, type of diagnosis, whether people have schizophrenia or not, has only 2 levels. So 3 by 2. There's 2 factors, one with 3 levels, one with 2. The total amount of conditions, by the way, would be 6. 3 times 2 equals 6. So that's a 2-way design. Here is an example of a 3-way design. Both of these are factorial. This is a 2-way factorial design. This is a 3-way factorial design. And so we have 3 factors. Factor 1 has 3 levels. Factor 2 has 2 levels. And factor 3 has 2 levels. If we were to calculate the amount of conditions here, it would be a lot. 3 times 2 is 6. 6 times 2 equals 12. So there's 12 total means that would be calculated in this study. So that's factorial nomenclature. Any questions or comments about factorial nomenclature? I've kind of already talked about these, so I'm going to skip over that. Here's just another example. So here's made up data. And I want us to, the reason I'm doing this example, it's like the example I just had with the bank robbery example, except it's a slightly more complicated design, because instead of only having 4 conditions, instead of only being a 2 by 2 design, this is a 3 by 2 design, so it has 6 conditions. And I just want to make sure that we're still comfortable with these concepts, even in slightly more complicated designs that have 6 conditions. And so the first thing I want us to do is look at the means that are compared for the main effect of amount of cups of coffee. So I have these numbers 4.5, 5.5, and 6.5. These are the 3 means that are compared for the main effect of amount of cups of coffee. And if you kind of look over here, you can see this column over here is everybody that has schizophrenia, whether they drank 0 cups, 1 cups, or 2 cups. Over here, this column is people that don't have schizophrenia, whether they drink 0 cups, 1 cups, or 2 cups. And we'll say, these are made up data, completely made up. We'll say that these scores in here are just short-term memory scores. So they take some test that measures how good their short-term memory is. That's the dependent variable. And that's what all these numbers in the columns mean. So our 2 factors are type of diagnosis and amount of cups of coffee. Our dependent variable are these scores in the middle, which we'll call something like short-term memory score. And then here are the means of each of our 6 conditions. For the main effect of amount of cups of coffee, there are 3 means that are compared. This one, and this one. Can someone tell me out there, why are there 3 means that are compared? In the previous example that I showed you regarding the bank robbery example, each main effect only compared 2 means. But now for this example, main effect of amount of cups of coffee, there are 3 means that are being compared. Can someone tell me why in this research design does this main effect have 3 means that are being compared instead of just 2?
There are like 3 levels: 0 cups, 1 cups, and 3 cups.
Yeah, 0, 1, and 2 cups.
Okay.
But yes, there are 3 levels to the factor amount of cups of coffee. And each one of these means right here corresponds. This is the mean of the 0 cups condition. This is the mean of the 1 cups condition. And this is the mean of the 2 cups condition right there. And so there are actually 2 ways of kind of calculating these numbers. The first way, which is the way I showed you earlier, though I showed you earlier in that table, is you can take the mean of people who have schizophrenia in the 0 cups condition plus the mean of people in the 0 cups condition that don't have schizophrenia. That 4 plus 5 equals 9. 9 divided by 2 equals 4.5. Another way you can get the same number, 4.5, which is to add up these 10 scores right here plus these 10 scores right here, and then divide by 20. And that would give you the exact same number, 4.5. The reason why oftentimes people will just want to compare these means right here is because it's just only 2 things to have to add together, which is, a little bit quicker compared to having to add 20 things and then divide by 20, which is a tiny bit more tedious. However, I'll point out once again, you can only use these means right here to calculate this number right here if the sample size of the two conditions are the same. 4 plus 5 equals 9 divided by 2 equals 4.5 gives you the exact same number as if you added up those 20 numbers and divided by 20. But those numbers will only be the same when the sample sizes of these two columns, the same amount of people are in each column right here. So moving over to this 5.5 right here, 4 plus 7 equals 11. 11 divided by 2 equals 5.5. Or you could just add up the 10 scores in this one column, then plus the 10 scores in this one column. That's 20 scores. Divide by 20. That would also give you 5.5. Finally, this number 6.5. 4 plus 9 equals 13. 13 divided by 2 equals 6.5. Or you could add up all 20 of the scores, divide by 20. That would also give you 6.5. So to make sure that we understand this, I want someone out there to tell me what are the means that would be compared for the main effect of diagnosis. Someone tell me what would be the two, there's two, I'm giving you a clue that there's two of them. What would be those two numbers, the two means that would be compared for the main effect of diagnosis?
Would it be the mean of this of one group than the mean of another?
Yes.
Like 4 plus 4 plus 4, and then the other one would be 5 plus 7 plus 9 divided by 3 for each of them.
Yes. So can anyone do the math? What would be the mean of these three numbers right here?
4.
Yeah, it's just 4. 4 plus 4 plus 4 equals 12. 12 divided by 3 equals 4. So that number right there, the mean score of people with schizophrenia is 4. And anyone good at mental math? What would the mean of these three numbers be? Anyone know?
7.
It would be 7. Let's see if, yep, and the individual in chat got it as well. And so 5 plus 7 is 12. 12 plus 9 is 21. 21 divided by 3 is 7. So this would be the main effective diagnosis, the mean score of people with schizophrenia compared to the mean score of people without schizophrenia. So that's just a little bit more practice for calculate. These are, once again, these are the marginal means. The marginal means are basically the means which are compared for each main effect. And remember that when you're calculating the means that are compared for each main effect, you're doing so by basically ignoring the factor that you don't care about. For the main effect of cups of coffee, we don't care about whether people have schizophrenia or not. And so we collapse across the two levels, schizophrenia and no schizophrenia, to get each of these means. And this analogous thing is true for the main effect of diagnosis. We don't care how many cups of coffee people drank. And so we just take all the people with schizophrenia, no matter how much coffee they drink, average all their scores together to give us 4. Same thing over here. We don't care how much coffee they drank. We just put all of the means together, all of the scores together, people, no matter how much coffee they drank, who don't have schizophrenia, and put them right there. Contrasting that then with an interaction. And these are, once again, I'll remind you, these are completely made up data. No study like this, to my knowledge, has ever been performed. And I'm not for sure it would be that theoretically interesting, because I don't think coffee has that significant of effect on people's short-term memory, at least that I'm aware of. But here's a way of visualizing the interaction. Once again, it's kind of similar to what I showed you on the previous graph. These are bars. This is like a flat line pattern in this cluster right here. I would call that like a flat line pattern. This is an increasing pattern. And so once again, the question I always ask myself is the pattern in this cluster different than the pattern in this cluster? And the answer is yes. I've actually done the statistical analysis in SPSS for this data. And yes, you do get a significant two-way interaction. But the key about interactions is understanding how to interpret them or what they mean. And what that means here is basically the amount of cups of coffee you drink, that doesn't influence your short-term memory score if you have schizophrenia. But if you don't have schizophrenia, at least according to these completely made up data, well, you tell me, if you don't have schizophrenia, how does drinking coffee influence your short-term memory score? Can someone tell me that? If you don't have schizophrenia, how does drinking coffee influence your short-term memory score? How do you describe, how does drinking coffee, short-term memory score is the dependent variable here. This cluster over here is the people that do not have schizophrenia. How does the amount of coffee that people drink, how is that influencing people's short-term memory when you don't have schizophrenia?
It's improving it?
Yeah, exactly. It makes it better. When you drink coffee with schizophrenia, it has no effect. There is no effect of cups of coffee here. But if you don't have schizophrenia, there is definitely a strong influence of amount of coffee right here. So there's a strong effect of amount of coffee for people that don't have schizophrenia, but there is no effect of amount of coffee for people with schizophrenia. So there's kind of, those are the two ways. The other way, once again, of saying this is a flat line pattern, whereas this is an increasing pattern. Those two patterns are different. Therefore, we have a significant two-way interaction. Questions or comments about this? We've got a couple more of these. And then, yeah, we'll get out of here probably in the near future. Here's another example of an interaction. So this is a good teaching example interaction. It does occur sometimes in real life, but it's a good teaching example because it definitely shows an interaction because there's the pattern here is a decreasing pattern. This is made up data once again, but I've changed the data. So for these made up data, if you have schizophrenia, if you drink, the more coffee you drink, it hurts your performance on short-term memory. So there's a decreasing pattern there. But if you don't have schizophrenia, the more coffee you drink, the more it improves your performance. So that's an interaction because that's a downward pattern. And this is an upward pattern. So the two clusters, each cluster has a different pattern than the cluster. But what's nice about this example is this is an example where there's a significant interaction, but there's absolutely no main effects. And in the way you, once again, determine whether there's a main effect is to calculate the marginal means. And you can do that here. If we wanted to look at the marginal means that were compared for the main effect of amount of cups of coffee, there's only two levels here. I simplified. The light bars are 0 cups. The dark bars are 2 cups. The way we would calculate those two means that are compared would be, this is the mean of, if we want to compare the two, the difference between 0 cups and 2 cups of coffee for that main effect. Basically what that means is we don't care whether people have schizophrenia or not when we're looking at the main effect of amount of cups of coffee. So we combine the scores of everyone that drank 0 cups of coffee, whether they have schizophrenia or whether they don't. And so the way you can do that here is by take this bar, this is a 0 cups bar for people with schizophrenia, 8, plus this bar right here, 0 cups bar with people that don't have schizophrenia, which is 4. 8 plus 4 equals 12. 12 divided by 2 equals 6. So that's the first marginal mean. Now let's go ahead. Well, I'll do this one, I guess, as well. 2 cups of coffee will do the same thing. So 2 cups of coffee, people with schizophrenia, that number is 4. 2 cups of coffee over here, people without schizophrenia, that number is 8. So 4 plus 8 equals 12. 12 divided by 2 equals 6 again. So the marginal mean for 0 cups of coffee is 6. That is the average score of people that drank 0 cups of coffee, independent of whether they have schizophrenia or not. We don't care if they have schizophrenia or not. That was 6. And it's the exact same mean as the people that drank 2 cups of coffee in this example, 6. And so if the two marginal means are the exact same number, 6, then we know there's not a significant difference. 6 and 6 cannot possibly be different from each other. So, and that shows that there's no main effect of amount of cups of coffee. And you could actually do the main thing with schizophrenia. You take that, average these two bars together, 8 plus 4 equals 12. 12 by 2 equals 6. Do the same thing over here with no schizophrenia. 4 plus 8 equals 12. 12 divided by 2 equals 6. There are no main effects. All of the marginal means are 6, which means that there are no significant differences for the main effects, but there is an interaction. So this is called hopefully I put it in the notes. I didn't, but I'll just tell you verbally that the crossover interaction is what this is called. It's an example. It's a special case of a study of results where there's a significant interaction, but there's absolutely no main effects. We get the interaction because the patterns of each cluster are different, but all the main effects kind of cancel each other out. So there's no significant main effects. Here's an example. This is a physiology course, and we'll learn a lot about event-related potentials in the second half of the course. And so I just wanted to provide, a sneak preview of the types of findings that we'll be reading about or learning about at least later on in the semester. So this is an event-related potential study. F3, this is EEG. The the EEG technology, which is basically measuring using electrodes that are placed on the scalp, measuring electrical activity, which is coming from the brain, but the electrodes are placed on the scalp. Here it's showing two of those electrodes, F3, which means frontal, and then 3, which is just a number system. But the 3 indicates that that electrode that's in the frontal lobe is in the left hemisphere. And then F4 is a frontal electrodes, which is over the right hemisphere. So we have these two different electrodes, which are measuring electrical activity, which is coming from the brain. F3 is right here. F4 is right here. So that's one factor in this study is location of electrode, left hemisphere or right hemisphere. The other factor, this is a study about music perception. A lot of my examples do come from that because that's what I know the most about. So this is a study where people were listening to a sequence of chords. It was basically a song. And then the last chord would either be like the correct chord for the song. This is the chord that sounds like it fits. Or it would be a deviant chord, which is like, you can kind of think of it as I'm playing on the piano and then that last chord, I screw up and play the wrong chord. And it sounds really off. It's like, one of those where it sounds bad. That would be the deviant chord. And what they want to, and the question is, in this study is, are the brain's responses to these chords different? And does the brain respond differently to these two chords depending on whether it's in the right hemisphere or whether it's in the left? Some of you might be familiar with the idea that music perception is occurs more in the right hemisphere than the left, which is for the most part true and supported by the ERP studies here. So in this particular study, they're interested in the ERP waves that occur right here, 180 milliseconds. So that this thing right here is time. This is when that chord is sounded. Either the deviant or standard chord occurs right here. Once again, or can occur right here. That's time point 0 when that chord occurs. This first dash line is 100 milliseconds after one of those chords occurs. And this is 200 milliseconds after the chord occurs. Once again, over here, 100 milliseconds after a given chord occurs, 200 milliseconds after the chord occurs. The standard chord, the chord that sounds correct, the pitches, the notes that sound good, correct, that fit with the song, those are the solid trace. Whereas the deviant chord, the chord that sounds bad, it's a messed up chord, the notes are wrong, that's the dotted or dashed trace right here. The question is, once again, does the brain differentiate, the electrical activity in the brain differentiate between these two chords? And does the brain's response depend on whether it's in the left or right hemisphere? And you can see that the responses are different. This 180 millisecond time point was where they focused their analysis. So right here, this is 180 milliseconds. It's slightly before 200. 200 milliseconds is right here. They're interested in this 180 millisecond time point right there. And once again, the 180 millisecond time point right here. And we're just going to talk about the interaction here. If you look at the, oops, where'd it go? I've copied and pasted the exact output, well, not the exact output, but I've some summaries of how these results were described in the results section of this study, Kolsch et al. 2001. And so you can read about that in your own if you want, but the basic idea is there's interaction here. Interactions on ERP data are a little bit more complicated to look at, but you can kind of, we'll have to get more, I'll teach you more about how to read these later on, but I'll give you an example of one right here. Basically, the difference between the standard and deviant in the right hemisphere is very big. So this is a big difference right here. Whereas the difference between these two waves in the left hemisphere is relatively small. So a big difference between these waves right here, a relatively smaller difference between these waves right here. This is the two-way interaction that was found in this study. And it supports this idea that music processing occurs more in the right hemisphere. The right hemisphere of the brain responds more to musical sounds than the left hemisphere. And that's true with a couple small examples, small counterexamples. The right hemisphere is more responsive to musical stimuli than the left. And you can see that here because the difference between these two waves is big, whereas here in the left hemisphere, the difference between those two waves is small. Okay. So almost done here, just a couple more things. We just read this for Monday's class. This was the Mozart Stroop 1. This is very close to the article that we're going to be, to the study that we're going to be doing for Group Project 1. And I think I literally copied and pasted the slide from last time. So I'm not going to spend too much time talking about it. I copied and pasted this from Monday slides. But maybe these slides will have a little bit more meaning to you now today that we've talked about what a main effect is. The main effective test session, the main effective condition, the significant test session by condition interaction. And I kind of explain what each of these things means. I'll focus right now on this test session by condition interaction, which you can see here. You look at this cluster right here, and you compare it to this cluster right here. And for the neutral cluster right here, I would basically say there does not seem to be an effect of the type of music. So remember, the type of music is this, whether it's consonant music, dissonant music, or silence. For the neutral condition, this is the Stroop task neutral condition, there's basically no effect of music, type of music, or what they would call, I think they would call that test session. No, they would call it condition. Yeah, the different types of music, they call it condition. So there's no effect of condition or type of music for the neutral condition, but there definitely is something going on in the incongruent condition such that when people are listening to the dissonant music, it makes their reaction times big compared to these other two conditions where the reaction times are relatively smaller. So this is like, I would call it like an inverted U pattern, whereas this is a flat line pattern. Those two patterns are relatively different from each other, so therefore there is a significant interaction. And then the article that we read for today, Sexy Stroop, which I've kind of summarized the main findings that I care about here. There's like 3 or 4 experiments in this study, but this is kind of the stuff that I review here is the stuff that I find to be most important about this article that you read for today. I guess a couple things. All the participants were heterosexual, about half were women, half were men. It was a factorial design, so going with the theme that we were talking about today. Technically, there are actually 3 factors. The sex appeal of the pictures, so some of the pictures that people looked at were low, that were not sexy. Some were high sex appeal pictures, that is the sexy pictures. The pictures that people were looked at were always of the opposite sex. Another factor was the biological sex of those people, the participants, whether they were female or male or women or men. And then the third factor was the congruency in the Stroop task. So whether the incongruent condition where you're naming the color of the ink, but the ink does not match what the color word says, or congruent condition where the color of the ink does match what the word says. That's the third factor. However, I should point out that they only analyze the data with a 2-factor ANOVA. They don't use the 3-factor ANOVA. And the reason is because they calculated a different score between reaction times in the incongruent and congruent conditions. This is what they call self-control measure. I asked you about it for one of the quiz questions, I believe. Self-control measure, this is the dependent variable right here. And this is a very, very important thing. They mention it here, I believe. The mean difference in reaction time between the incongruent and congruent trials. Each of these bars represents the difference between the incongruent and congruent trials. That's what each of these bars mean. They're all positive because everyone scored, or on average in each condition, people scored more slowly in the incongruent condition than they did for the congruent condition. Basically every single condition that was true, people were slower for the incongruent Stroop condition than the congruent Stroop condition. The self-control measure was a different score, incongruent minus congruent. And because they did that, that means that they did not have to include congruency as a third factor in the ANOVA. They could just look at these 2 factors in the ANOVA, and they don't actually care about the factor congruency, which is a little bit sketchy of a thing to do, actually. There's reasons mathematically why it's not that big of a deal. Personally, if I were doing this study, I would not have calculated the different scores. I would have done the 3-way ANOVA because it's just slightly less sketchy to do it. What they did was a little bit sketchy. It's not a textbook thing to do. So they did find a significant interaction. That's the really crucial finding. That's what a lot of the questions I was asking you about were today. And you can see that interaction right here. Remember, each of these bars represents the difference between the incongruent and congruent conditions. That is what the researchers called like Stroop interference. And what they wanted to know is for men and women participants, Does Stroop interference depend on whether the pictures that people had just looked at were unsexy or sexy? And they found the interesting finding, at least I found it interesting, that when men, that's the dark colored bars, looked at sexy pictures, that caused a lot of Stroop interference. Stroop interference, once again, is measured by if you have a very big difference in reaction times between the incongruent and congruent conditions, as if you're really slow in the incongruent condition compared to the congruent condition, then you're going to have a very tall bar right here. And that was particularly true for men after they had just looked at sexy pictures of women. But at least in this study, when you took the women participants and they looked at sexy pictures of men, they showed a significant Stroop effect, but it was significantly smaller than the Stroop interference effects that the men found. There's a big difference between the bars here. Men in this high sex appeal, they are men looking at sexy women, showed a lot of Stroop interference compared to women that were looking at the sexy men right there. So men had more Stroop interference after looking at the sexy images compared to women who are looking at sexy images of men. And if you look at men and women over here, it's not that just men have worse Stroop interference overall, because if you look at the low sex appeal condition, whether you're a woman or a man participant, basically there was not a significant difference here. And so I want you to always connect this. There was a significant two-way interaction. How do we kind of think about this, conceptualize it? I would say there's basically a flat line pattern here. These two bars, they're slightly different, but they're not significantly different. So it's basically a flat line pattern here compared to an increasing pattern here. These patterns are different enough that you would call it a significant interaction. They did do post hoc tests, which are what the bracketed lines are showing here. This post hoc test, this top bracket, was significantly different. There's a significant difference there. This post hoc test right here, the second bracket, was not significantly different. So this is the last thing I'll show you here, which is this is the last quiz question. Everyone got credit for this on Blackboard. Everyone got credit. But this was the question: which of the following best represents your opinion of this article? You were surprised by the findings and you find them believable. 6 people said that. I was surprised by the findings. I found them unbelievable. 4. I was not surprised by the findings. 6. And then people that were kind of indifferent. They could not say they're surprised or not. There was 6 right here. So this is the column. 6, 4, 10, and 6 were the amount of answers here. And I just wanted to point out the great amount of heterogeneity, the great amount of differences in opinion regarding this article. About half the class was surprised by the findings. The other half of the class was either not surprised or kind of indifferent to the findings. And I just wanted to point that out. I just found it interesting, the amount of heterogeneity regarding people's opinion regarding this article. So once again, these are the things that we need to take care of. We do have some time before class period's over. I strongly suggest you use this class time to finish up some of these things. And if you have questions about some of these things, feel free to ask me. I'll hang around my computer. I'll be answering emails until 3:45 pretty quickly. So go ahead, go to it. And let me know if anyone wants to have a Zoom meeting. I'm happy to do it. Also, if you have not formed groups yet, and you are here and you want me to help pair you with other individuals, I can use breakout rooms to help you guys find meeting rooms real quick. Otherwise, take care, and I will see you Friday for lab.
I still need a group.
Okay.